Hello everyone, and you're very welcome to today's CPD Professional Subject Knowledge webinar for the Healthcare Science T-Level. And the focus of today will be for the content from B1 around genetics and genomics, before moving on to the B2 content, which focuses around the applications of genomics within healthcare science. The plan for today's session is to begin with some quick introductions first. We'll have a brief overview of the T-level qualification. We'll then look at the B1 section of the specification before looking at B2 regarding the genomics and its application. And finally, to address any questions that you may have. But please, throughout this webinar, do continue to put questions into the questions box of the GoToWebinar control panel, as we will be stopping throughout this webinar to address them. And this session is also being recorded, and you can access the slides that's being used for this webinar from the handout section of the GoToWebinar control panel. But first, I'd like to begin just with a little poll to gauge your current confidence with the teaching, delivery, the content, etc. for this section, for the genomics and genetics section. So what you'll see on your screen right now is just a little poll just to gauge your confidence, but 1 being little to no confidence and a 10 being very confident. So just put where you think you are at the minute. So we've got about a third of the votes in, and it's split evenly at the minute, 1 to 2 and 3 to 4. So just a few more votes to come in yet. I'll just give about 10 more seconds or so. We've got about 60% of the votes in. Looks like most people are in the 3 to 4 bracket in terms of the confidence with this at the moment. Okay, just going to close off that poll. As we can see there, most people in the 3 to 4, no one going into the mid-range, so all in the lowish end in terms of confidence. Hopefully today's webinar will help you and make you feel a bit more confident going forward with the requirements of this content, etc. So first of all, just a few quick introductions. So my name is David Kiernan, and I work in the provider development team at NCFE. And I support centres with the delivery of our qualifications, the structure, the assessments, and anything related to teaching and learning. And my direct email is shown there on the screen, but if I'm unavailable, then do please use our general provider development email address. And I'm also joined today by Joe, who'll also briefly introduce himself.
Hello everyone, my name is Joe Neem and I'm the Science Subject Specialist here at the NCFE, meaning I support with science queries across the business and our various qualifications. I also support with the delivery of the T-level in healthcare science, the structure and anything related to teaching and learning. My direct email address is also shown there, and back over to you, David.
Cheers, thank you very much, Joe. So what we're going to do now is just for anyone who's brand new to the T-level qualification, we're just going to do a brief overview of how it works, the assessments therein, etc. If you want more comprehensive information, then do please see one of our onboarding webinars. So what is a T-level? They are a level 3 2-year program aimed at 16 to 19-year-olds, and they are equivalent in size, rigor, and depth to 3A levels, and do attract UCAS points as well. The T-levels are owned by the Institute for Apprenticeships and Technical Education, or IFATE, and the entire design and content for the T-levels came from employer panels in the relevant industries. And because of this, the T-levels do offer students a route to paid employment, but with UCAS points being available as well for the completion of a T-level, students can progress onto higher education or onto a higher level apprenticeship. And the entire design of the T-levels is to ensure students have the threshold competency for the relevant occupational role. And that's the most important aspect, ensuring students have that threshold competency. The specification itself can be divided into 2 sections, the core component and the occupational specialism component. So the core component can be further divided into 3 sections. Section A, which is working in the health and science sector. Section B, which is the scientific knowledge, concepts and understandings of biology, chemistry and physics, and then the core skills. So this core content and skills form the foundation that the occupational specialism content and skills is built upon. Now, some of this core content is also what's called root core content, meaning it will be common to the health, science, and healthcare science T-levels. So the Section A root content are is also root content. Then we have the content and knowledge specific for an occupational role. And there is just one occupational specialism that's live as of now, as of September 2021, but others are planned and are in development. So the content, knowledge, and skills will be different from one specialism to another because it's all about having, again, that threshold competency for the job role. And there is one other part of the technical qualification that I want to highlight, which is the general competency framework. And this lists the English, maths, and digital skills competencies that students will also be taught. And again, the focus is for students to have that threshold competency for the job role. These competencies are assessed and are embedded within the specification too, but it would be useful to develop these further with your students. And the T-level assessments can be broken down into the core assessments and the occupational specialism assessments. The core assessments consist of 2 written exam papers covering the core content in Section A and Section B, and an externally set skills assessment called an employer set project, so called as the tasks had input from employers to make them as realistic as possible. The occupational specialism is assessed in year 2 with externally set synoptic assignments, which are also designed to be as occupationally realistic as possible. I'm now going to hand over to Joe, our subject specialist, who's going to go through the genetics and genomics content.
Thanks, David, and hello again, everyone. So as previously mentioned, we're going to be looking at genetics and genomics, and we're going to start off with the genetics content of B1. Throughout the session, there will be a Padlet open for you to refer to the specification points we will be covering. This will allow you to add any comments or ideas that you may have about the initial planning and delivery of genetics and genomics. It can also be updated with any ideas that pop up throughout the session to ensure we are sharing that good practice amongst ourselves. There's also a section at the start for any general concerns that will help us with future support and planning. Please be aware though that this is just for general concerns around any other specification sections that you may have and want covered in the future. Any questions that you have that are specific to this webinar and its content, please make sure you pop into the question box. The link for the Padlet will now be going into the chat box, and I'm just going to give us a quick 30 seconds just to ensure we can access that.
So everyone should have access to that link. It's been popped into the chat, so you can click on it there. And there's the Padlet on the screen now that you should be able to see.
Perfect, and please do make us aware of any issues you may have with accessing the Padlet via the chat box. So the first point at which genomics is mentioned is within the specification at B1.19, the difference between genetics and genomics. Now this is the last specification point within the genetics section of B1. And generally when interpreting the specification, the point in bold is the overarching learning point, and then any bullet points and sub-bullet points are the depth in which students need to understand. Now the depth provided here is specifically just to highlight the difference between genetics and genomics, with genetics focusing in on the functioning and composition of single genes, whereas genomics focuses on the entire genetic material of an organism, including coding and non-coding DNA. Now this follows on from information provided around DNA, RNA, and their role in inheritance within B1. So although this is the first mention of genomics, in order to fully understand its applications within B2, It's beneficial to go into more detail within B1 genetics in order to help equip students with the knowledge they need. Now, this isn't required by the specification, but it will be invaluable to support students' understanding of the various applications of genomics they will go on and cover within B2. So as an initial example, within B1.13, it would be useful to expand on the depth provided here just by introducing the term genes. Quite simply, this can be done by expanding on the first bullet point by saying that DNA holds genetic information within short sections known as genes. And although not directly mentioned at this point, transcription and translation would also be useful to mention here, as they are referenced in functional genomics in B2. Now, I don't mean to cover transcription and translation in masses and masses of detail. However, this point can be expanded by saying that genes are within DNA, are transcribed into RNA. That RNA then travels to the ribosomes within the cytoplasm, where it is translated into proteins. You may wish to include some information on mRNA to better contextualize this to students. However, they just need to know that genes code for proteins, and this process occurs via sections of DNA, aka genes, being transcribed into RNA and then translated into proteins. Within B1.14, there are certain sections again that can be expanded upon to help students with their understanding. So where it talks about the component of the DNA nucleotide, mentioning the bases can then be linked back to expand the definition of genes. So just by explaining a gene is a specific sequence of bases. The structure of DNA is then referenced as being a double helix containing two polynucleotide chains. This can be linked back to the definition of genome, as DNA polynucleotides vary from 50 million nucleotides all the way up to 260 million nucleotides within the human nuclear genome. This can then be linked back to chromosomes, as humans have 23 pairs, each containing these DNA polynucleotides, resulting in the human genome itself containing approximately 3 billion base pairs. However, it only consists of 20 to 25,000 genes. So this information can just be used to reinforce the fact that the majority of DNA is actually non-coding. And understanding this will ensure students understand the definition of genome in reference to coding and non-coding DNA. Now, I know that's a lot of information, so just to summarize the content from those last few slides. In order to help students understand and apply genomics in B2, they will need to be aware of what is meant by the entire genome. So using humans as an example, our cells contain 23 pairs of chromosomes within the nucleus. Each chromosome is made up of DNA, as shown by the diagram there. A small coding section of DNA is known as a gene. However, in between these coding regions are large regions of non-coding DNA. Now, all these coding regions and non-coding regions within each chromosome make up the entire genome. And in terms of the coding sections of DNA, these are known as genes and code for proteins. And this process starts, again, as shown on screen, with transcription, where DNA is transcribed into single-stranded RNA. RNA then travels to the ribosomes within the cytoplasm, where it is translated into a protein. Finally, for B1, through covering the process of semi-conservative replication within B1.16, students should understand that the process ensures genetic continuity between generations of cells. However, sometimes mutations occur, which in the specification is referenced as a spontaneous change in the DNA sequence, which can lead to genetic variation. Now, this is important to highlight as this is the basis of evolution. Now, when a mutation leads to a change in a characteristic that results in that organism being better adapted to its environment, this organism is then likely to go on, survive, reproduce, and pass that characteristic on to the next generation. And when this happens over many thousands of years, it leads to the process of evolution. And this process of evolution has led to an estimated 8.7 million species of plants and animals alone on this planet, all with different genomes, which vary amongst different genomes, which then vary amongst the species themselves, with each organism having a different set of genes. Again, humans can be used as an example of this with students, so with students within the class having different eye colours and so on, just as an example of genetic variation. Now, the reason I've gone into so much detail there about evolution, even though it's not required, is it's important to highlight and potentially cover because evolutionary genomics then appears within B2. Now, just because I've thrown a lot of information there at you, we're just going to take some time to review the Padlet and share some good practice around B1 genetics. You might already have some ideas of the depth you've covered. So please add any of those ideas into the relevant section. If you have any good sources of information for any topics, please feel free to share these also. And we're also going to take this opportunity to check and answer any questions in the chat. So there's no questions so far, but feel free to add any into the question box into the chat if any do pop up. Just give us another couple of minutes as I can see people typing away onto the Padlet. The typing does appear to have stopped, so I'll just give us another 40 seconds or so just to see any comments come through. Okay, so no comments have come through as yet. Feel free to have this on in the background and add in as we're talking through certain subjects. That's absolutely fine. We will be reviewing it later on in the session. So we're now going to move on and look at the specific applications of genomics within B2. And you'll be able to see where the learning within B1 and the points that we've covered really supports this understanding within B2. And we'll be discussing content depth and some delivery ideas as well. So before going into the specific applications of genomics, B2.20 first secures that understanding of genomics and how it's different to genetics. So here it introduces a specific definition of the genome, which you may now have already covered in B1. And here we also expand on the definition of genomics, and you could give some examples here in terms of different genomes. So this could be anything from a human genome all the way to the genome of a butterfly. That's just to secure the fact that the term genome does not just apply to humans, but all organisms. And here it would be good to mention DNA sequencing methods to the students. They don't have to know the methods in detail, but again, you can link that and use that learning from B1. So quite simply, it's the methods that sequence the specific order of bases in DNA and therefore can be used to map the genome, as well as the specific order of bases in coding regions known as genes. At this point, don't worry too much about talking about bioinformatics because this is covered within a later section within the specification. So here are some activities taken from the scheme of work. In terms of delivery, present the students with the definitions of genome, genomics, and genetics. And as an activity following this, students as individuals, then as pairs, then as larger groups, and then as a class agree why the concept of genome and genomics is different to genetics. However, there is also a chance here for a quick formative assessment task by presenting students with the definitions along with the keywords, and then just a quick match-up activity just to check that students can recall the understanding of those keywords you've covered. In terms of spaced repetition, this task could then be copied into later sessions just to test that recall of these keywords and to help with the embedding of that knowledge. And there we can see the answers for anyone who was testing themselves there. Moving on to B2.21, the characteristics of different study areas within genomics. Now I've split these into different areas of study just so we can analyze each in terms of depth and how it may link to previous learning. So here we're starting with structural genomics, which is the study and description of the three-dimensional structure of every protein encoded by a specific genome. This is one of the reasons why I suggested it was a good idea to introduce the idea of transcription and translation in B1, just so that students are fully aware that genes code for proteins. However, this can also be linked back to the learning within B1.7, which talked about functional proteins such as fibrous proteins and globular proteins containing a number of polypeptide chains, which will determine the shape and size and function. So in terms of bringing all that information together, coding regions within genomes, known as genes, code for proteins, and these proteins will have a three-dimensional structure, aka its shape and size, which will determine their function, and all of which is coded for by that specific order of bases within genes. And obviously applying the definition from the previous slide, making clear that the genome includes all the genes for an organism. The next study area is functional genomics, which is the study and interactions of all genes and therefore proteins in a cell. It focuses on dynamic aspects such as transcription, translation, gene expression, and protein interactions. Again, this is another reason why I'd suggest covering transcription and translation within B1, as it saves you having to go back here and reteach it in more detail. Instead, it will just be a recap of learning. Here, there's the opportunity also to go into large amounts of detail around transcription, around translation, gene expression, and protein interactions. But this is not required in terms of the specification. So here I would start with gene expression by highlighting that the body is made up of many different types of cells. Each of these cells, however, will have the same set of DNA and therefore the same genes. But different types of cells will have different genes within their genomes that are expressed. Because of this, only the expressed genes are then going to be transcribed and translated into proteins within those cells. And that's the link to transcription and translation within the specification point. To contextualize this to students, if they're still not very clear, highlight to them that although every cell in their body will have the same set of DNA, except reproductive cells, as they have the half the number of chromosomes, their skin cells will behave in a very different way to, say, their white blood cells. Yet within their nuclei, they will have the entire same set of genetic information. Now, these are different cells because some genes are expressed and some aren't. Basically, some genes are switched on and some are switched off. And that's all the detail you need to go in. For protein interactions within this point, link this again back to that previous learning around transcription and translation. If different genes are switched on and off, that's then going to lead to different proteins being present within the cell, and therefore different protein interactions. If your students are interested, you could discuss how some proteins are responsible for gene expression. However, you may want to keep it at a level that is not going to confuse them to a point where they no longer understand. And next, the specification talks around evolutionary genomics, which is the changes that may occur in a genome over an evolutionary timescale. Next is epigenetics, which is the study of DNA modifications, which do not change the DNA sequence, but can affect gene activity. Now, because you may have talked about evolution in B1, as discussed, that means you won't have to spend too much time on this. It's just quite simply comparing the genomes of different organisms over that evolutionary timescale, and it's used to invest what organisms other organisms have evolved from. Epigenetics can be linked to gene expression, also covered in the last slide. So quite simply, the modifications of DNA structure that affect gene expression, aka which genes are switched on, which are switched off, but do not change the specific sequence of bases. Now, DNA modification causes these genes, again, to be switched on or off, and therefore affect the protein composition of the cell. And again, that's all the detail you need to go into. Students do not need to have knowledge of, say, DNA methylation or histone modification, just quite simply that modifications of DNA structure can affect which genes are expressed. So after all that information, here are some input ideas in terms of delivery, again taken from the scheme of work. And it suggests a group activity where the group is split into 4 groups and each investigate a different area of genomics and then come up with an A4 infographic. However, in terms of my personal opinion, I would actually start with making sure that there's some tutor input around those areas of study that we've covered, just so students have that clear understanding of each area and the depth in which they need to understand. After this, they can then complete the research activity as described into each study area of genomics. And as an extension, you may even want them to research the Human Genome Project and explain its importance within genomics research. This is just an extension idea and it's not something they need to be aware of, but just in terms of a potential activity or even a home learning task. I've also provided some links there to sources of information around genomics, and a positive of those links is that they don't go into too much detail that then may confuse students. And these links will be available in the handout. Now on this slide, we can see an example worksheet that could be given to students. So quite simply, students are given a section for each area of study within genomics and the option of an extension task at the bottom. Again, you could get students to start off with a summary of each study area based on that tutor input. This would just ensure that they've got those clear definitions to refer to when revising, and then allow them to research those specific areas after this. In terms of formative assessment for B2.21 then, you could have another match-up activity like before, which again could be embedded throughout various sessions to ensure that they are clear on what each area of study focuses on. You can use example assessment questions, which can be created using the specification. And just as an example, here's one I came up with, just based on looking at the specimen exam questions and then using the specification. So just quite simply explain the difference between functional genomics and structural genomics. So using the information that the students will have covered, the answer will be functional genomics studies the functions and interactions of all genes in a cell and therefore all proteins. Functional genomics focuses on dynamic aspects such as transcription, translation, gene expression, and protein interactions. Structural genomics, on the other hand, is the study of the three-dimensional structure of every protein encoded by the genes within a specific genome. Now, because that's just a comparison between the two definitions given in the specification, this would be assessing an AO1 skill, which is students demonstrating their knowledge and understanding of context, concepts, theories, and principles in healthcare science. And that AO1 skill, as shown on the screen there, counts for 26 to 32% of the core component exam papers. Now, just to break up the session, we're now going to have another example of how the specification points can be used to form assessment questions for you to have a go at. So the example on the screen here is what dynamic aspects would feature in the study of functional genomics. So is it transcription, translation, gene expression, anaerobic respiration, aerobic respiration, protein interactions, functions of genes, binary fission, big long list there. We're just going to give you a minute to access the Slido and answer. And at this point, we'll also check to see if there's any questions in the chat. So we've had a comment to say that there was an interesting article on the BBC website about gene silencing today in the health section that could be shared with learners. Yeah, brilliant. If you've got a link to that, that would be brilliant to put in the chat or even on the Padlet, just to share so everyone's got access to that. That's brilliant. Just give us 30 more seconds just for any more comments or questions or queries. Brilliant, thank you. We've now got that link, so that the link to the article has been posted in the chat there by David. Thank you for that. Thank you for that comment.
Thank you, Dan, for sharing that. Actually, very good article. Great to see that good practice being shared. I might actually add it into the Padlet as well.
Yeah, that's brilliant, Dan, and that's brilliant to highlight as well. Thank you for that, Dan, because there's always news stories. So it's always good when you're coming up to planning these topics just to double check news outlets, because there's always very interesting stories around this topic. So moving on then. So yeah, I think it wasn't quite clear that there was multiple options here that you could have picked. That's my fault, sorry. So the correct answers being transcription and translation, gene expression, protein interactions, and functions of genes. Those are just the ones from the specification point there. And these short AO1 questions can be very useful as a review, starter, and home learning tasks to help embed the retention of those key facts. Now that we've gone through some examples of interpreting depth of the specification, I will be moving slightly quicker through the following points, just highlighting key details as well as some delivery ideas, just to make sure we keep to the hour. For B2.22, how techniques in genomics are used to investigate, diagnose, and treat disorders. It begins with gene sequencing, which is used to detect the order of bases within DNA that makes up the genome, which then obviously corresponds to the proteins that will be produced. It does mention here codons, but again, don't fear that you have to go into a massive amount of detail. You can contextualize this just by quite simply explaining that a codon is 3 bases which code for an amino acid. And again, linking that back to the B1 section on proteins, students should be able to link that to amino acids being the building blocks of proteins. Then the application of gene sequencing is it will identify if a particular cell is unable to produce a specific protein for physiological function, such as an enzyme needed for a metabolic reaction. Now, this is very important in identifying inherited diseases, and an example within healthcare could be identifying mutations within the cystic fibrosis transmembrane conductance regulator, or the CFTR gene, which can lead to cystic fibrosis. Again, that's not required, nor is going into detail about needing 2 copies of that recessive gene, but it's just an example of the application of gene sequencing. So here, genome mapping and genome editing are presented as techniques within genomics. The importance of genome mapping is where it's used to identify and record locations of genes on a chromosome and the distance between genes. It will also identify any specific chromosome abnormalities, and again, just in terms of contextualization, you could cover monosomy conditions such as Turner syndrome, where there's 1 chromosome instead of a pair of chromosomes, or trisomy conditions such as Down syndrome, where there are 3 chromosomes present instead of a pair. And then for genome editing, it's used to change the DNA within a cell by adding or removing DNA sections or altering the DNA sequence. This can enable a genome to produce proteins which it was not able to make previously, or affect the non-coding sections to switch on or off gene expression and protein production. Now, this can be, as another example, could be used to edit or replace a mutated CFTR gene, linking it back to the example from the previous slide. However, there is an opportunity here to potentially debate and discuss with students and link back to section A 5.12, which covers the importance of ethics and research ethics in the healthcare science sector. And again, this links to the comment by Dan, providing that link. There'll always be news stories around gene editing and its potential, and therefore you can get students to debate whether they think genome editing is something we should be doing, whether they agree, whether they disagree, and their reasons why. B2.23 then just goes into the applications of genomics within healthcare science. And in terms of delivery, the depth needed is provided on screen. So as an example, through gene sequencing, genomes can be mapped, and this can be applied in the example shown. So during prenatal tests during pregnancy to screen for specific genetic disorders due to family history or other factors. Again, cystic fibrosis being an example consistent with that, with what we've discussed in the previous sections. Now, the scheme of work presents delivery ideas for B2.22 and B2.23 specification sections. It suggests that students should research the techniques used in genomics, so gene sequencing, genome mapping, and genome editing, and then share their research with the class. They would then go on and link that to the applications provided in B2.23. It also says that the tutor might indicate some suitable websites for students to avoid wasted time. And so to help with this, I've just provided a few links there just to direct students to some valuable sources of information. And again, if you've got any others, including news links and things like that, please pop them onto the Padlet, which we'll be reviewing shortly. And I'm just going to take this opportunity just to double check that no questions have come through and to answer any that have. So another comment by Dan, there's a good question on this within the sample assessment material. Fantastic, so yeah, please do go on and have a look at our sample assessment material as well. One, to help you build your own questions, but brilliant that there's a good example of this already there, and that can be accessed via the healthcare science page on QualHub. Okay, so if we're going to move on now onto the next section, which is the application of genomics within B2.24. And we're going to discuss the importance of bioinformatics within the area of genomics. Now, I've split this section up just to make sure, just to make some key points, again, around depth and delivery. So first of all is an introduction to bioinformatics and the fact that it brings together biology, computer sciences, mathematics, and statistics. It's used to organize, analyze, and understand biological data sets to make the data understandable and clinically actionable. Now, this can be linked back to the earlier points I was making about the fact that the human genome is approximately 3 million base pairs long. Furthermore, the original Human Genome Project ran from 1990 to 2003, but we can now map a human genome in a day. This is due to advances in the techniques previously discussed, such as gene sequencing, but then also due to the use of bioinformatics software. So using this contextualization of the massive size of the human genome, mapping the genome and genes results in a massive amount of data. And it's through using bioinformatics software that we can organize and analyze this biological data extremely quickly. And this rapid analysis means the outcomes are clinically actionable in a short time frame. So this section talks about the role of developing software for the analysis of biological data. And again, building on the idea of the size of the genome, it's safe to say that it's working with very big data. So using larger data sets, which is referenced for more accurate results, is actually more of a mathematical application. But this can be linked to genome-wide association studies. For example, and the example it gives, the 100,000 Genome Project, which is a project by the government to map 100,000 genomes of people with inherited diseases and with types of cancer. The aim of that project is to compare those genomes to identify genes that may directly or indirectly cause those diseases. In order to do that, we need the bioinformatics software that relies on statistical analysis to compare those genomes rapidly. And due to that bioinformatics software, this results in that automation of analysis, which results in much faster analysis, and there's fewer manual tasks involved. Also, another application of bioinformatics is it can be used for prediction modeling. So for example, I said that there's certain genes that can be indirectly linked to certain diseases. So if a patient is identified as having a certain composition of genes, this may make it more likely for them to develop a certain disease or even cancer. For someone who is likely to develop a disease based on this testing, so for example, dementia, this could lead to early identification and treatment. Now, in terms of delivery ideas, the scheme of work suggests tutor input around the importance of bioinformatics within the area of genomics, and students write up their notes from the sessions in their file. But again, if you wanted to turn this into a bit more of a research task, I've included some links there to provide the students with some more information. Specifically, there's a link there to the 100,000 Genomes project by Genomics England. And in terms of a case study idea as well, you could get students to research the BRCA1 and 2 gene and their links to the development of breast cancer. Through genome mapping and through the fast analysis using bioinformatics software, a woman can have their genome sequenced and the BRCA1 and 2 gene can be identified and can be used to model prediction in terms of how likely it is they are to develop breast cancer. We're now going to review the Padlet just for a few minutes and share some good practice around genetics. So again, if you've got any more news articles or any more links or any just general delivery ideas around any of these topics, please do pop them into the Padlet. If you have any good sources of information, okay, whether it's news, whether it's a revision website, whatever it may be, feel free to pop those in. We're also going to check any questions within the chat. I'll just give us a few minutes just to go on and add any specific comments onto the Padlet. Any ideas, any news articles, feel free to share. Also remember, as we're heading towards the end now, feel free to add anything into those general knowledge concerns or queries that will help us then plan future webinars around any general knowledge concerns and queries.
What I'll actually show on here as well, just as Dan quite nicely shared this BBC article. I did put the link to the chat, but here's the article that Dan did share.
Brilliant, thanks David. And what I'll do is I'll just go through a question that was sent beforehand. So question here, do students have to know specific applications of genomics? So for example, why individuals would need to be screened as an example of family history. So that's more going back to B2.23. And in response to this, specific examples, as I've said throughout, do not need to be covered, but students do need to be able to apply their learning. So we discussed the AO1 skill, AO2 skill, which makes up the highest percentage of marks within the core component papers of 40%. So for example, students could be presented with the following questions. So a woman and her partner are carriers of the mutated cystic fibrosis transmembrane conductance regulator, CFTR gene. She is currently pregnant and wants to know if her child will have cystic fibrosis. Suggests what tests can be carried out. So just applying those tests from B2.23, there would be two options. So prenatal tests during pregnancy, which would screen for the specific genetic disorder of cystic fibrosis during the development of the fetus, or screening the infant when it's born, and both would require gene sequencing in order to identify the mutated CFTR gene. And this is just an example of an application of learning question that could be used. But again, just in terms of that question that's sent in, don't need to know specific examples, but they do need to be able to apply that learning to different situations. If it's okay, David, if you could just share that link with people as well. So just as another example of potentially fueling your debate and your discussion, there's just going to be a link to a New Scientist article around CRISPR. Now that is something heavily involved in genome editing and gene modification. It's quite simple, okay? It finds specific genes and it can swap them, replace them, turning genes on or off, seen as quite revolutionary within genetic modification, and that really relates back to that learning about genome editing and the possibility of gene editing, and it can really fuel that debate with students around whether this is something we should be doing, what are the benefits of it, and what are the ethical concerns relating to being able to edit genes. So if you just type in CRISPR, it'll come up with many different articles. This is just one around its applications.
Yeah, I've put that link into the chat, so hopefully everyone here can see this. Just want to address another question that has come in just regarding a request for training on writing T-level exam questions. That'd be some useful CPD. Thank you. In fact, at the end of this webinar, there is going to be an evaluation form that comes up, and we do want you to put in anything in there as well, any future things that you may want to see. It is something that I do speak about as well, as it often comes up during my one-to-one consultations with providers about how to use the specification to generate questions. So that's certainly something that would be discussed as well, that can be discussed in those calls. But do please, those CPD suggestions that you might have or any other things you may want in that evaluation, if you pop that in there, that would be brilliant.
So we'll give a couple more minutes for any more. If any more questions pop up, please feel free to add them in. Anything on the Padlet, feel free to add. Obviously, we'll be staying on if you wish to add any more questions or following on using the email addresses at the start of the handout. Please feel free to send us questions directly, and I'll now hand back over to David.
Cheers, thanks very much for that, Joe. Yeah, as Joe mentioned, do please keep any questions you have coming in. But I would like to just check in terms of confidence now as well, just like I asked at the very start, where would you put yourself now at following this? So again, 1 being little to no confidence, 10 being extremely confident. I'm not expecting anyone to be in the 9 to 10 bracket, probably yes, until you get the delivery. That's where people usually start to find, but just where do you think you are at the moment? Okay, so our votes are coming in. It looks like we got all the votes in, that's excellent. Some people have moved on, not everyone, but we do have 50-50 at the minute, 50% in the 5 to 6 bracket. So we didn't have anyone in the 5 to 6 bracket. That's good. Still have some people though in the lower end, 1 to 2. Please, if there's something specific, this is the chance. We want you to pop that into the question box so that we can best support you. Now we are going to remain on this webinar as well for the remaining few minutes so that we can address any of those questions that you may have that are going to come through. If there's anything that you would like to ask and check, this is definitely the time to do it. However, if you don't have any questions, do by all means feel free to leave. And there is going to be, as I mentioned a moment ago, a little evaluation that is going to pop up. And your feedback is very important to us. It is just some multiple choice questions and then one final option for any additional comments you would like to make. And if you are leaving now, from everyone here at NCFE, we would like to wish you a very safe and successful year.